Human musculoskeletal anatomy. We've got a little calculation first. The stage micrometer and eyepiece graticule, they've labelled them. I don't think they'd actually look like this. I've never seen an eyepiece graticule where you can't see all of it down the microscope. Anyway, use a diagram to calibrate the eyepiece graticule. And what we've got, I need to line these up. So the zeros are lined up already. And then usually what we do is find the 100 on the eyepiece, but we can't do that. So we just need to line this up somehow. And we go so many eyepiece units are so many stage micrometer units, and so on. So here's one version of the calculation. So we can see 80. You could have 82 eyepiece units is 100 stage units. One in the mark scheme actually uses 80 and 98. Let's do it that way around first then, shall we? So 80 eyepiece units, 80 eyepiece units equals 98 stage units. One eyepiece unit is that, and we know that each one of these is 10 micrometers, or so each one of these units, as this is that's 0.1 millimeter, and that's 0.01 millimeter. So each one of these, so one stage unit is 0.01 millimeters, or 10 micrometers. So times by 10 there, divided by 80, and that should give us 98 times by 10 divided by 80 is 12.25 micrometers. An alternative thing that you might do here, you might do it the other way, and you might go 82 eyepiece units of 1 millimeter. That's probably the way I'd do it. Looking at the diagram, 82 eyepiece units equals 1 millimeter. So one eyepiece unit is 1 millimeter divided by 82. So 1 divided by 82 equals 0.012 millimeters, and I could convert that by moving the decimal place 1, 2, 3, 12 micrometers. That's the same thing, just another way around. Using the calibrated eyepiece graticule, section of bone was viewed, and the diameter of the Versian system shown below was measured and calculated to be 450 micrometers. All very interesting. Why would it be necessary to recalibrate the eyepiece graticule at higher power objective lens? So if we change this lens, then the eyepiece will stay the same, but the stage will be more zoomed in. You'll be able to see less of it. So you need to recalibrate it again. So the eyepiece unit has different values for each calibration. Different values for each calibration at each objective lens. In the space below, draw the structure shown above as it would appear in longitudinal section. So this is a transverse section or a cross section. So this is kind of like a circular. So what we can see up there is that, but it's like that. So we want if we chopped it, took a section of there and chopped it in half lengthways like a banana split, what would we see? So you'd see this canal here, and then these lacunae sort of going like that, and then the whole thing would be enclosed. So something like that. But it's just trying to see whether you know what a longitudinal section is. Many of the cells in the lacunae have well-developed rough endoplasmic reticulum Golgi bodies. So now here in the core knowledge, rich in RNA, I think it's giving you enough clues there to figure out that you're talking about protein synthesis. And we need 2 marks there. So what's the point in protein synthesis? Well, they must make a protein, for secretion outside of a cell or something like that. So for release or secretion from cells. Then here we are. Describe the transport system by which oxygen and nutrients are supplied to these cells. Well, in here, in these canals, we've got arterioles and venules. We've got blood supply and nerves and things like that. So that's what we are needing there. So we've got arteries and veins, or arterioles and venules. So arteries and veins, where are they? They're in the Versian canals. If we were drawing this, so that's the sort of diagram that we started with. You get these other canals that go sideways as well. Those are what they call Volkmann canals. Volkmann's canals. And tiny little fissures with capillaries in, and those are canaliculi. So we've got capillaries in canaliculi. Singular canaliculus. So that's how nutrients get to the cells. The important thing there is the cells, not just how they transport in the middle. And now we're going to talk about rickets, and they are linking rickets as an increase, but something to do with decrease in milk consumption and reduction in outdoor play. So milk, we're going to relate that to diet, and outdoor play we're going to link that to UV light. So that's the information that's the path it's trying to send you down. And so your job now is to go sort of, lack of milk in the diet. What's in milk? So that would be, like calcium in milk, e.g. less calcium. Let's talk about less outdoor exposure to UV light or ultraviolet light for production of vitamin D in the skin. So you don't absorb vitamin D from sunshine, but it helps you make it. And then the link between the two, so vitamin D is needed for absorption of calcium. So vitamin D is needed for absorption of calcium from the gut. Describe the role of calcium ions in the contraction of skeletal muscle. So this is just a bit of theory. Now muscle contraction, just let it all flood out. So calcium ions released from the sarcoplasmic reticulum, they bind to troponin, but you might not say that, which troponin changes shape. The important thing really is that this moves tropomyosin away from the binding sites and actin. So moves tropomyosin away from the actin binding sites on myosin. So that's the key thing. And then you get the most important thing, which is then cross bridges form. And then possibly you know it's not in the mark scheme, but you might talk about the role of calcium ions in synapses, on the way to in nerves on the way to skeletal muscle, possibly. Last page. When you die, your muscles remain in a contracted state, rigor mortis. Why? Well, if you're dead, you're not respiring anymore, and respiration provides ATP to make these cross bridges. So energy from ATP needed to release these cross bridges, or to break cross bridges. And with death, no respiration, so no ATP available or synthesized. Part E, reasonably straightforward if you know your difference between the fast and slow twitch fibers. And slow twitch fibers we associate with endurance sports. So we'd probably go for person B there, the endurance athlete, and the person with the most fast twitch fibers, person C, for the sprinter. So B, endurance athlete, C, sprinter, and then we need to explain our answers. So endurance athlete, large, what's the difference between these fibers? So large numbers of mitochondria, myoglobin, aerobic, those sorts of arguments. Fatigue slowly. Whereas the sprinter, we associate that with anaerobic, quick contraction, strong force. So the opposite of what we've set up there.